This tutorial will use PlasmoDB to outline the features of a gene record page. Gene record pages on all UPathDB sites are structured similarly, so anything you learn here can be applied to other sites. To reach a gene record page, either enter the gene ID in the quick search box at the top of the page or click on the ID in any result table. Let's navigate to the gene record page of P. falciparum dihydrofolate reductase by clicking the ID in the result table of this text search. Each gene record page is an expandable list of all the data in the database for the gene. The present view shows the list collapsed. Clicking Show next to the data track title displays the data track. Click the Data Sources link on the right to access information about the origin of the data. Show all or hide all data tracks at once using the links in the upper right corner. The Download link allows you to save the gene record on your computer. The sections of a gene record page include the Overview, Annotation, Protein, Expression, and Sequence sections. You will need to scroll down to view all the sections. I'll go through these one at a time, pointing out some highlights. The gene page opens at the Overview section, with the gene ID and product name at the top, followed by some useful links. Let's open the Genomic Context graphic. DHFR-TS is highlighted in yellow in the top track of the graphic. Its location in the P. falciparum genome is shown in relation to the surrounding genes with the chromosomal scale shown above. Below that are tracks showing the same region for the other species and strains. The gray lines between the tracks represent synteny. Mousing over any gene in the diagram opens a pop-up with information about the gene and some useful links. Let's collapse the genomic context graphic and consider other data tracks in this section. The next data track, Regions or Spans Associated by eQTL, displays a table listing the haplotype blocks that contain this gene and other useful links concerning these loci. This table, comparison to previous release, lists all the changes in annotation that have occurred since the last release of our database. The Multiple Sequence Alignment track is a quick link for running an alignment of the gene against other database genomes. Let's scroll down to the Annotation section. The Annotation section begins with a link for adding your comments about the gene to the record. Comments appear in the second entry as a table. The External Links track opens a table of useful links such as the GeneDB, Entrez, and UniProt records. The table of names, previous identifiers, and aliases can be very useful when cross-referencing with other information sources. Now let's look at the Protein section. The Protein section begins with a graphic showing features of the gene's protein product, similar to the Genomic Context graphic. The graphic displays data such as proteomics peptides and interpro domains. Some tracks are empty because no data from the track maps back to the gene. For example, since no peptides found in this Scheizont experiment mapped to the gene, this track does not contain data. The proteomics data is also available in a table. These two tables provide links to the protein databank and immune epitope database entries for this gene. Let's scroll down to the expression section. This section contains data about expression of the gene's transcript. So in this case, the graphs and charts revealed by clicking Show represent the subset of data from the experiment that applies to the DHFR-TS transcript. Data is arranged by experiment, and links to information about the data sources are opposite the data track title. Expand the first option to view the pattern of DHFR-TS expression during the erythrocytic stage as determined by microarray over a 50-hour time course. The data is available as a table, and more than one style of graph is often available for display. The Sequence section contains the genomic, coding, and predicted protein and mRNA sequences. These links are good for a quick cut and paste. You can download the sequences in FASTA format from the Overview section. Thank you for watching this tutorial. Please contact us with any questions or suggestions.